How can the clinical laboratory detect carbapenemases, which are enzymes that can make bacteria resistant to some of the most potent or broad-spectrum antibiotics available? What's the clinical significance of detecting such an enzyme? Does it affect the care of the patient? Finally, what practical advice can we give labs to help them decide which of the many available tests for carbapenemases is the best one for them? Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org. I'm Alex McAdam, your host and editor-in-chief of JCM. This podcast is supported by the American Society for Microbiology, which publishes JCM. We're talking about detection of resistance to the carbapenems, such as imipenem and meropenem. As mentioned, these are among the broadest spectrum antibiotics available. Bacteria that are resistant to carbapenems were identified as urgent threats by the CDC in 2019. Some bacteria produce enzymes which destroy these antibiotics, and these enzymes are called carbapenemases. By producing a carbapenemase, bacteria become resistant to the antibiotics, making carbapenems ineffective for treatment of infections caused by such bacteria. I'm joined by an expert guest to discuss detection of carbapenemases, Dr. Patricia Simner. Dr. Simner is an associate professor of pathology at Johns Hopkins University and director of the medical bacteriology laboratory there. She's also a newly appointed editor of JCM. Dr. Simner wrote an outstanding review in JCM called Phenotypic Detection of Carbapenemase-Producing Organisms from Clinical Isolates in 2018. It's a few years old, but I think it's still the best available overview of these tests. We'll also talk about one of her more recent articles later in the podcast. Trish, thanks for joining us.
Thanks for having me, Alex. I got to say, I'm excited to be a new JCM editor as well. I've been really enjoying reading some of the submissions coming into JCM.
That's great. I appreciate that very much, and I think you're doing a wonderful job with it.
Thanks.
I want to start with a basic question. Why is it important to detect carbapenemases? For example, does the result determine whether the laboratory reports the patient's bacterial isolate as susceptible or resistant to carbapenems?
That's a great question, Alex. So carbapenemases are important for multiple reasons, and the detection is important mainly for two reasons. The first is for patient management. It has treatment implications because carbapenemase detection, when you detect a carbapenemase, and specifically the class, it might have implications on the type of treatment, as some of the new agents that have activity for carbapenemase producers have activity against some carbapenemases, but not all. And so, for example, ceftazidime-avibactam can have activity against class A or your KPC producers, and class D, OXA48 type enzymes. Whereas meropenem-vaborbactam is more specific and has activity against class A, or more specifically for KPC. And then none of these new agents have activity against the metallo-beta-lactamase producers. Furthermore, some of these new agents might be reserved specifically for carbapenemase-mediated carbapenem resistance over other mechanisms of resistance, which could include ESBL and DAMP-C, with poor mutations in efflux, and so it might have stewardship implications as well. And so the first importance is that carbapenemase detection has implications for treatment. The second reason why hospitals may want to detect carbapenemases is for infection control reasons. And this could be just to understand the epidemiology of carbapenemases that they're experiencing. And so most labs in the US have encountered KPC producers before, but it's also good to know when you encounter non-endemic variants that come into your hospital system, where more strict infection control enforcement may be required. They might actually have patient and provider cohorting, additional cleaning of rooms when carbapenemase producers are detected, and specifically if it's something new to the system. And so, for example, at Johns Hopkins, we actually have different regulations around infection control and contact precautions on whether our carbapenem-resistant Enterobacterales is a carbapenemase producer or not. And so if it's a carbapenemase producer, those patients are automatically flagged and put on contact precautions. Whereas if we encounter a non-carbapenemase mediated carbapenem-resistant organisms, such as an Enterobacter, which we might see more commonly being non-carbapenemase mediated. If it doesn't meet our MDR flag, then those patients might not be on contact precautions. It would be the carbapenemase production that would flag those ones to be on contact precautions. So there's multiple different steps in terms of infection control that might be put in place based off of carbapenemase, off the detection of carbapenemases. But coming back to your question, does this impact the way we report our carbapenem AST results? So currently, if you're using the most up-to-date CLSI guidelines, using the post-2010 carbapenem breakpoints, then you should be reporting the carbapenem results as tested. And the carbapenemase detection is not really required for reporting of your AST results, but is more required from, as I mentioned, an epidemiologic or infection control standpoint, or for patient management and treatment purposes. That said, for treatment purposes, it's most important in labs that may not have access to AST devices or antimicrobial susceptibility devices for some of these new agents, where the carbapenemase detection and perhaps the particular class can help you out with deciding what's maybe one of the novel beta-lactam, beta-lactamase inhibitor combinations you might want to use for treatment. But you should always confirm the results by AST testing to ensure that it does have activity against those carbapenemase producers, because just because they're new agents doesn't mean they have guaranteed activity against these carbapenemase producers.
So let me see if I've got it right. So useful for predicting susceptibility results, but those should be confirmed by phenotypic testing. Useful for infection control practices, but generally should not be used for altering reports from susceptible to resistant or vice versa, because those should be based on direct susceptibility testing.
Correct. But the only scenario that you would want to use carbapenemase tests for directing or reporting your AST is if you're using the old carbapenem breakpoints.
The pre-2010.
Pre-2010. And unfortunately, there are still a lot of labs that are using the old obsolete breakpoints, which they should make it a priority to update to the most current breakpoint, because it has patient safety implications as well as public health implications, where, for example, you might not detect a carbapenemase producer because those breakpoints are higher, and it might go spread unknowingly in your institution if you're still using those higher breakpoints. And so that's why if you're using the old pre-2010 breakpoints, you should be, if you encounter an Enterobacterales that has an MIC, an imipenem or a meropenem MIC of 2 to 4, or an ertapenem MIC of 2, if this looks suspicious for carbapenemase production, and you should be performing a carbapenemase test. And if it's positive, then you would modify your results to resistance. But that is only if you're using the old obsolete breakpoints.
Got it. Thank you, Trish. And just for listeners, Enterobacterales is what we used to call, it's the family that we used to call Enterobacteriaceae, and let's not open that can of worms. But you can just, listeners can mentally cross out Enterobacterales and write in Enterobacteriaceae, and they will be all set. You touched on this a little bit, but can you give us a basic framework for classification of the carbapenemases? I think that'll be useful in some of the things we're going to talk about.
Yeah, so it's important to note that carbapenemases are beta-lactamase enzymes. And so beta-lactamases are classified by the Ambler classification scheme, or most commonly classified. And this is based off of amino acid sequence. And so based off of amino acid sequence, beta-lactamases are classified into class A, B, C, and D. Now the carbapenemases themselves fall into class A, B, and D. And furthermore, they can be broken down into whether they're serine carbapenemases or metallo-beta-lactamases. Now, the serine beta-lactamases or carbapenemases have an active site serine, and those include your class A, which would include your KPC producers, or your class D, which are your oxa-type enzymes, which include the OXA48-like enzymes, or some of the oxa enzymes that we encounter in Acinetobacter. Whereas metallo-beta-lactamase producers are your class B enzymes, and these require zinc at the active site for activity, and those include your NDM, your VIMs, and your IMP. And the requirement for zinc at the active site is also exploited for detection and separation of serine and metallo-carbapenemases in the lab.
That's great, thank you. And we may come back to that if we have a little bit of time for it. Before we dive into the different tests for carbapenemases, which we'll come to next, can you first tell us which bacteria should be tested for carbapenemases? What should trigger that testing, and which genera or species should be tested?
That's a great question, Alex, and it's a question I encounter often when I'm presenting on carbapenemase testing. And so I think the most important group that we should be testing for carbapenemases is in the Enterobacterales. And the reason being is that they are heterogeneous in their mechanisms, where, for example, at Hopkins, it's about a 50-50% carbapenemase producing to non-carbapenemase mediated mechanisms of carbapenem resistance. Now, that might not occur at every institution, and it may vary based off your based off of the epidemiology, but it does become important within that group of organisms because there's no easy way of knowing whether it's a carbapenemase producer or not based off the AST profile. So the most important group are Enterobacterales. But the other question is do we want to test it in Pseudomonas aeruginosa or Acinetobacter baumannii? Well, for Pseudomonas aeruginosa, the main mechanism mediating carbapenem resistance is non-carbapenemase mediated. It's mediated through porin mutation, and more specifically OPRD mutations, in conjunction, with other mechanisms such as AMP-C and efflux. And so for Pseudomonas, especially within the US, I don't think it's of value to be testing for carbapenemases because you're not really getting the bang for your buck. It's very rare. So within Canada and the US, the rate of carbapenemase production among carbapenem-resistant Pseudomonas is around 2%. And so unless there are instances that you may want to consider testing. So if you're in an area of the world where the epidemiology is different, where you actually see a lot of more carbapenemase production in Pseudomonas, that is definitely something you might want to consider. If you're experiencing an outbreak at your institution, then you might want to consider detection of carbapenemases in Pseudomonas. And then this one time I've actually we've done it, or sometimes we do it in the lab here, is if we see an unusual AST profile. And what do I mean by that? Well, the most common carbapenemase harbored by Pseudomonas are metallo-beta-lactamases. And metallo-beta-lactamases are really good at hydrolyzing most beta-lactams except for estrianem. And so once we had a urine culture from a patient from an endemic area where they see a lot more carbapenemase producing Pseudomonas, and the AST profile was completely resistant except for estrianem. I'm like, I wonder if this is a metallo-beta-lactamase producer. And so we had, when we verified our phenotypic test, we had already verified it for Pseudomonas, and so we tested it out, and sure enough, it was a carbapenemase producer. We did further phenotypic testing, or genotypic testing, and it turned out to be a VIM producing Pseudomonas. And you can't rely on estrianem, but especially because multiple times these metallo-beta-lactamases produce other beta-lactamases that would usually provide resistance to estrianem, but that's kind of a hint that you could take for when you might want to be suspicious of a carbapenemase in Pseudomonas. Now, going to Acinetobacter, it's the complete flip side, where the great majority of carbapenem resistance mediated by in Acinetobacter is mediated by carbapenemases. But the carbapenemases that are produced by Acinetobacter are usually confined to Acinetobacter. And these are oxa-type enzymes, not the traditional OXA48-like that we think of in the big 5 carbapenemases, but these are your OXA23-like and your OXA40-like enzymes that are normally confined. So if you're testing for carbapenemase production in Acinetobacter and a carbapenem resistant strain, it's most likely going to be positive if you have an accurate test. But there are some instances that you might want to consider carbapenemase testing in Acinetobacter, where last year we got a letter from our state health lab saying that there has been a recent outbreak of NDM producing Acinetobacter within the state. And so we wanted to see if we were encountering this. Now, a broad-based phenotypic test wouldn't be helpful because we know it should be positive because they are oxo-type enzymes that are produced as well. But if you would want a more specific test that would identify the genotype there, where we were using a more specific molecular type test to detect whether NDM was present or not in Acinetobacter. So in short, it's really important in the Enterobacterales. And in the US and Canada, not so important Pseudomonas unless you're experiencing an outbreak. And then for Acinetobacter, again, not so important because we know that the majority of resistance is mediated by carbapenemases that are usually confined to Acinetobacter.
Thank you, Trish. You mentioned phenotypic and genotypic testing. Can you just give us a description of those two broad categories, and then we'll dive into each of them in a little bit more detail?
Yeah, so phenotypic tests are tests that actually detect the activity of the carbapenemase either through hydrolysis tests or detect the phenotype associated with carbapenemase production through some sort of growth-based test. So an example of that would be kind of an e-test strip looking for a particular phenotype associated with carbapenemase production. Whereas molecular tests detect the gene that encodes the carbapenemase enzyme.
Thank you. So let's dive into the phenotypic tests. And I think the one that most people who have heard of phenotypic tests for carbapenemases, the one that people are likely to be familiar with is the modified Hodge test. Can you explain how that test works?
Oh, the good old modified Hodge test.
I never liked it.
I remember the first time I performed it. So the modified Hodge test is probably one of the first and the most well-known carbapenemase test. So if you recall, the good thing about the modified Hodge test is that it uses standard reagents. It's cheap, that are available in most labs. So you're taking a Mueller-Hinton plate, you're streaking a lawn of a completely susceptible E. coli that specifically is carbapenem susceptible. And then in the middle of that plate, you're placing a carbapenem disc. Now that can be an ertapenem or a meropenem disc. And then you're using that disc as your hub. And then to test your organism of interest, you're going to streak that carbapenem resistant organism that you're trying to figure out, is this a carbapenemase producer or not? And you're going to streak it away from the carbapenem disc towards the edge of the plate. So kind of a hub and spoke type thing. And on each plate, you could put multiple organisms, whether you're querying whether they're a carbapenemase producer or not. And then you're going to incubate that plate overnight. And if you didn't have those organisms there, you would see your standard zone of inhibition around that carbapenem disc for that completely susceptible E. coli. But because you have those organisms streaked away from the disc, that if they're carbapenemase producers, they'll actually allow the local concentration of carbapenem to be lower in proximity to the streak. So you'll see the carbapenem susceptible organism growing in towards the disc and kind of, forming this kind of clover leaf shape morphology and indentation towards the carbapenem disc. Along that streak line, if you see that, that means that your organism that you streaked was a carbapenemase producer. If you don't see any indentation along your streak, then it's a non-carbapenemase producing organism. Now, the problem with the modified Hodge test is it has both sensitivity and specificity issues. So from a sensitivity standpoint, it's not very good at detecting metallo-beta-lactamase producers. Whereas from a specificity standpoint, it's actually a lot of ESBL and AMP-C producers with porin or efflux mutations would actually produce false positive results. The other thing is it can be very tricky to interpret and can be quite subjective. And so for that reason, it's really kind of fallen out as a go-to carbapenemase test. And CLSI actually in the last couple of years removed the endorsement of the modified Hodge test as an acceptable carbapenemase test.
So easy to do, or at least uses available reagents, but not very sensitive for some important carbapenemases, and not very specific because AMP-Cs and ESBLs can cause false positives in the presence of porin mutations. So good riddance to it. We had such trouble with it in my laboratory. Every time we would do it and we'd send an isolate out to the CDC for confirmation, they would say, no, it's not a carbapenemase producer. So we mostly had specificity problems with it, but let's leave it in the rearview mirror. So that leaves us with two major categories of phenotypic tests that depend on bacterial growth to detect the carbapenemases. So let's start with one of those. Let's start with the Karba NP test. Can you tell us how that one works?
Yeah, the Karba NP test was first described by Drs. Nagma and Poirel, hence the NP. And now these are hydrolysis-based tests. So what you're doing is you're taking a bacterial extract. So you're taking your organism of interest, that's a carbapenem-resistant organism. You're going to lyse it and take that extract, and you're going to put it in the presence of an imipenem-based solution with a pH indicator, most commonly phenol red. If the organism is, and you're going to incubate for 2 hours. So if the organism is a carbapenemase producer, then that organism, the carbapenemases that were extracted and released will then hydrolyze that imipenem in the solution, lowering the pH, producing a yellow color. So you're going from red to yellow if you have a phenol red, indicating a positive carbapenemase test. If it's a non-carbapenemase producer, then within that 2 hours, they're not efficient at they're not going to be efficient at hydrolyzing that imipenem, and so that tube will remain red, and you'll have a comparator tube without imipenem to monitor your color change. And so that's how the Karba NP test works. Now there's advantages and disadvantages. The biggest advantage to the Karba NP is that it's a rapid test. It's a 2-hour test. And it actually has, better performance characteristics than the modified Hodge test. Except for, unlike the modified Hodge, the weak spot with the Karba NP is the OXA48-like enzyme. So it doesn't have really good sensitivity for OXA48-like enzymes. It also can be a little bit subjective. Again, you're looking at a color change. And so you could see color changes from red to orangey color. And so sometimes it could be subjective in whether you want to interpret. So kind of like what you were saying with the modified Hodge test, but not to that extent. And then you could also have issues with mucoid isolates with the Karba NP. Again, you're relying on that extraction process and having enough carbapenemase within that extract to cause that color change to occur. I think, this was one of the tests that I tried bringing in because I was super excited that we had a better test than the modified Hodge test as a carbapenemase detection test for clinical care. But my techs were kind of dragging their feet about implementing it. And it's because you have to make up reagents. And the imipenem solution was only stable for up to 3 days. And it can be quite wasteful if you don't have high volumes. You're making up a solution that you might use a little bit of to set up a few Karba NP tests, but if you're not going to use it again within 3 days, you're going to be throwing that out. And so it was a whole lot of work. And so was something we chose not to implement in the end, but it could be the advantages of the Karba NP are those is the rapid turnaround time. And so many commercial manufacturers are like, hey, we can get around the disadvantages of this test. And so some manufacturers, such as Biomidier, came out with the Rapidec Karba NP, which is a lyophilized strips with all the reagents. So you're not making up those reagents anymore and overcoming the concerns that my techs had initially, or the techs here had initially in our lab. Or there's the Roscoe Diatab kits, which are essentially lyophilized reagents in a diatab that will dissolve once you reconstitute it in solution. So there are some commercial manufacturers that have overcome the kind of bigger hurdles to implementing a Karba NP type test, a manual version of that test in the lab.
And you mentioned it's less sensitive for which ones?
OXA48 like
OXA48 like enzymes. Can you remind us which organisms are most likely to produce those so people will know what to watch out for?
Yeah, OXA48 we see in our Enterobacterales more. And, you know, OXA enzymes together are actually, as a class, are just not very good at hydrolyzing carbapenems all that rapidly. And so the same with, so this is another reason why Acinetobacter, some of these tests don't perform well with Acinetobacter, because, as I mentioned before, the main enzyme is oxidase, and they don't hydrolyze the carbapenems that rapidly and easily. And so, or efficiently, I should say, and that's the reason why they have lower sensitivity for detection of some of those enzymes.
Got it. So it's the rate or the efficiency of the hydrolysis that makes that a weakness for some of the tests, but exactly. The OXA48 like enzymes are still sufficient to confer resistance to the organism.
Exactly. Yeah.
Got it. All right, let's go on to the next test, which is the MSIM test. First, what does MSIM stand for? And then can you tell us a little bit about how the test works and its advantages and disadvantages?
Well, Alex, I really wish I could tell you that the MSIM stands for Simner, but that is not the case. Unfortunately, it stands for the Modified Carbapenem Inactivation Method. Now, why modified? Well, the initial version was described as Carbapenem Inactivation Method, and then a CLSI working group went and evaluated it as another carbapenemase test to perhaps endorse and use for clinical labs. And we modified it slightly compared to the initial version, hence modified carbapenem inactivation method. So how does the method work? So this method is an overnight method where you're first taking your carbapenem resistant organism of interest. You're taking a loop full of that organism, a 1 microliter loop for Enterobacterales, a 10 for Pseudomonas aeruginosa. You're incubating it in 2 mLs of TSB broth with a meropenem disc. And you're going to incubate that at 35 degrees for 4 hours. Now, that 4 hours is important because if it's a carbapenemase producer, it will be very efficient at hydrolyzing the carbapenem within the meropenem within that disc within the 4 hours. If it's a non-carbapenemase producer, it's not as efficient at hydrolyzing it within that 4 hours. And so the meropenem mostly retains its activity. So how do you assess whether it has activity or not? Well, you're going to pull that meropenem disc out of that broth that you had your organism interest in, and you're going to swab a lawn of a Mueller-Hinton plate with a susceptible indicator strain in E. coli, usually your E. coli 25922, and then you're going to place that meropenem disc onto that plate, and you're going to incubate overnight. And then the following day, you're going to look to see if there is a zone of inhibition or not. If there's no zone, that means that meropenem, or a little zone, that means that meropenem was completely or almost all hydrolyzed by that carbapenemase producer in that broth that you incubated it with initially. If there still remains a zone, so that would be positive. If there still remains a robust zone greater than 19 millimeters, then it would be negative for carbapenemase production because the non-carbapenemase producer was not able to hydrolyze the meropenem within that 4 hours and still has activity against that indicator strain on the Mueller-Hinton plate.
Thank you. And we should give a little shout out, I think, to the Clinical Laboratory Standards Institute, or CLSI. And I know you're a very active volunteer with CLSI. This group, which is, again, staffed by volunteers, it puts out fantastic guidelines, very useful guidelines, and really in terms of susceptibility testing, I think we would be lost without them. I want to turn now to the last phenotypic tests, which are the lateral flow immunoassays. And you were an author of a paper in the July issue of JCM titled Evaluation of NG Test Carba 5 for Rapid Phenotypic Detection and Differentiation of 5 Common Carbapenemase Families, Results of a Multi-Center Clinical Evaluation. So let's use that as our example of lateral flow immunoassays. So to begin with, how does that test work?
Well, these tests, unlike the phenotypic tests that I've been describing so far, actually detect the enzyme itself. And how it does this is through a lateral flow assay. So similar to a home pregnancy test, where on the nitrocellulose membrane, they have antibodies, monoclonal antibodies that are specific to the carbapenems that are carbapenemases that they're trying to detect. So for the NG test Carba5, it actually has 5 different strips, and each strip has monoclonal antibodies that have been immobilized on that strip that are specific to KPC, NDM, VIM, IMP, and OXA48. So each strip is specific to each one of those carbapenemases, and those are the big 5 carbapenemases. And so if you have a sample, you'll take an extract and you'll put it into the well, and that'll run up the nitrocellulose strip. If the carbapenemase is being produced or the enzyme is present, it'll bind to those monoclonal antibodies. There'll be a second set of antibodies that flow as well that will bind as a sandwich and produce a color, creating a strip if that carbapenemase is present. So you can have one or multiple strips positive if it's a carbapenemase producer. We've shown it's got pretty good activity if you got let's say an OXA48 and NDM co-producer. And you'll also want to look to make sure that your control band is demonstrating a valid test as well. And so I was very excited about the introduction of this test because it's rapid. It's 15-minute test. It's something that's easily worked into the bench workflow. And you're not only able to detect the carbapenemase, but you're also that it's a carbapenemase producer, but specifically the enzyme being produced. So you're actually looking at expression of an enzyme in these isolates. So I was very excited about the introduction of these tests.
So before we talk a little bit more about your study, I just want to make sure that we've got it. It's a phenotypic test in the sense that it's detecting a protein, something produced by the bacterium, rather than a gene for that protein. But it's different from the phenotypic test we've already discussed in that it's not detecting the activity of the carbapenemase. It's detecting just the presence of the carbapenemase using antibody binding to the carbapenemase. Is that correct?
Yeah, you got it.
Got it. All right, good. So tell us about the study. Tell us what you and your collaborators did, how you did it, and what you found about the test.
Yeah, it was a really fun study. A great group of individuals participated. Dr. Lars Westblade, Dr. Steve Jenkins, Nate Lenenbauer, and Carrie-Ann Burnham. So it was a really fun group to work with. And so we participated in a multi-center clinical trial so that they can obtain 510K or FDA clearance of the diagnostic test for use within the US. And so what we did is we took 309 Enterobacterales and Pseudomonas aeruginosa isolates, a total of 309. These were both prospectively and retrospectively collected isolates. And we compared the lateral flow, the NG test Carba5 results to a composite reference standard. And why is that important? Well, it's important in just what you had just summarized, is that these tests detect slightly different things. So the lateral flow antigen is detecting the protein, the enzyme being produced. And so we needed a reference standard that we can compare to that would allow us to detect a similar thing. So the composite reference standard included the expert Carba-R molecular test, which is a molecular test for the big 5 carbapenemase genes. So you're not actually detecting expression. So that's the reason why we also needed to include the MSIM test as part of the comparator to look that not only is there a carbapenemase gene present, but it's actively being expressed and we could detect the carbapenemase activity. And so that was served as our reference standard. We also compared the LFA from blood and McConkey agars and found really good sensitivity and specificity. After discrepant analysis, it was 100% concordance. So it was really good. And then that was at 3 sites. And then a 4th site at Dr. Burham's lab, she had actually compared blood, McConkey, and detection from Mueller-Hinton agar of the lateral flow, and again in comparison to the the molecular Carba R test and the MSIM, and found really good agreement again of the lateral flow from the various media types. Why is that important? Because you might be taking an isolate from different media for confirmation of or for detection of carbapenemase production. And then the Mueller-Hinton agar is interesting because if you're a disc diffusion lab, it would be really cool that once you read your AST results, that you could also pick some colonies from around that disc, especially if it's a carbapenem resistant organism, and run that directly on your lateral flow immediately to detect whether it's a carbapenemase producer, or I should say, whether it detects one of the big 5 carbapenemases. And so that was one of the additional studies that were performed. And so overall, it was looked really good.
I happen to know that Dr. Burnham's lab is a disc diffusion lab or KB lab, so I can see why they would add that into the study. I think in the clinical microbiology world, we appreciate how important it is to validate things off all different types of media. It can seem like a trivial thing, but can be very important for workflow in our laboratories, so that makes sense. I want to turn now to the last sort of group. I'm going to treat this group broadly, which is a little unfair to the group, but that is the genotypic test for carbapenemases. And as you mentioned, these detect the genes for the carbapenemases rather than detecting hydrolysis of the carbapenems or detecting the enzymes themselves, like in a lateral flow assay. So can you compare, just in general, the utility of these tests to the phenotypic tests that we've been talking about?
Yeah, so molecular tests are nice because you don't actually need the growth of the organism to set up, like with the phenotypic tests. And so these molecular tests can be especially helpful for detection of genes directly from clinical specimens. And so what we've seen is on these syndromic-based panels, whether they be respiratory or from positive blood culture broths or direct from whole blood, they've been putting some of the major antimicrobial resistance genes as markers on these tests. And that could be extraordinarily helpful for patient management and treatment implications if you detect a carbapenemase producer from a positive blood culture broth rather than having to wait 2 days later or directly from a respiratory specimen. So the detection of them are definitely helpful. So and can have treatment implications, most likely escalation of therapy. However, the absence of them can be tricky, especially in conveying that result to your clinicians, that the absence does not mean that they're going to be susceptible, because as we've discussed, there's many different mechanisms that can mediate carbapenem resistance. And so I think they are definitely very helpful direct from clinical specimens because they're rapid. And the problem is that they can be quite expensive. And that's why, from an isolate standpoint, most labs do phenotypic tests because they're just a little bit more affordable than a molecular test from isolate testing. The other scenario that these molecular tests can be helpful with are for surveillance purposes. So gastrointestinal colonization of carbapenemase producers are a major concern and contribute to the spread within the hospital system if they're unknowingly colonized. And so some labs or institutions do surveillance for testing for these. And so those molecular tests can be helpful because you can get a same-day results. We've validated the expert Karba R for eSwab testing for detection of the big 5 carbapenemases, but there's also the BD check CPO assay that is FDA cleared from rectal swabs for testing the big 5. So there's also surveillance aspects to those. And then the last application, as I kind of alluded to, was you could also use these molecular tests, such as the Cepheid Karba R, directly from isolate testing as well, and get the similar results to the LFA of the big 5 carbapenemases, but with a heftier price tag and the requirement of an instrument purchase upfront as well.
Thank you. It does sound like a pretty expensive proposition in terms of surveillance testing. That
Yes,
if you were doing widespread surveillance within your institution, it would
not widespread.
carry a big bill.
Yeah.
So we've talked about all these different carbapenemase tests, phenotypic, genotypic, of all different variety. Which of these tests are you actually using at Johns Hopkins, and what specific applications do you have for them?
Great. So we'll start out with isolate testing. We actually have a algorithm that we follow based off of the AST profile. And again, we only do carbapenemase detection in Enterobacterales on a standard basis. And so if the organism is intermediate or resistant to ertapenem only, we'll set up the modified SIM, the MSIM, and have results the following day. If the isolate is resistant or intermediate to both ertapenem and meropenem, then we automatically, we just switched over to the Carba5 lateral flow assay to get same day results out for patient care. The reason is that the mono ertapenem resistance, you can kind of weed out a lot of the non-carbapenemase producers by running the overnight test and not using. We used to use the Carba5 test, or the Carba R. I'm sorry, I keep getting those two. The molecular Cepheid Carba R test, and so we felt like, oh, we'll use a more affordable test for those that are mono resistant to ertapenem first, and then if they're resistant to both, we'll use the more expensive test.
So I'm sorry to break in, but just to pause there for a second. So if it's ertapenem resistant only, but susceptible to meropenem, it's unlikely to be a carbapenemase producer. And so you use a less expensive test to weed them out, as you said. Do I have that?
Yes, in a sense, yes. So they're less likely to be a carbapenemase producer,
Gotcha.
not unlikely, because we actually do see quite a few KPC producers that are only ertapenem resistant, but for the most part, our enterobacters that are mono ertapenem resistant, for the most part, are non-carbapenemase producers that we encounter. And so it was just kind of a step that we took when we were using a more expensive diagnostic for detection of the big five upfront to kind of weed those out.
That makes sense. And it sounds like that would cover most or perhaps all needs. Are there additional carbapenemase tests that you're using in your lab?
Yeah, so we run the GenMark gram-negative panel from our positive blood culture broths for gram-negatives, and so we do have the big five carbapenemases are covered on that panel. So we will only report those out when positive and not when negative. And again, I kind of alluded to that earlier, was because we don't want physicians to have false hope that when they're not detected that they're going to be carbapenem susceptible. But when those genes are detected by the panel, it is important that they know, because they're going to escalate therapy in those scenarios.
And do you have some piece of text so that people understand when you say there's a carbapenemase present, that they understand what the significance of that is for selecting treatment?
Yeah, so we have antibiotic guidelines that coincide with the results from the GenMark panel that help, but we actually decided not to do a very wordy templated comments in our resulting, but there are EPIC notifications pop-ups that come up that if the patient's not on the right therapy, there might be a suggestion this patient should be on this treatment. So let's say not a carbapenemase, but like CTXM's detected, a pop-up will come up and say this patient should be on meropenem. So we actually use more of those prompts because we find that those are more likely to be acted upon than a very long comment in our results. We also have a very active stewardship team, which we're very fortunate to have here. And so they will be following up on a lot of these results that are flagged to them in EPIC to follow up to make sure that they're on the right therapy.
I think that's great. So the comment isn't don't use this, don't use that. It's something much more proactive. This is what should be used.
Yes.
That's great. That's great. I think
Yeah.
you'd have to have a strong stewardship team in place to do that, and I'm sure you work with them in developing those comments as well. But you cannot underestimate the value of antimicrobial stewardship and micro working together.
Exactly. Yeah, we have a great team.
Anything else to add? Anything that we've missed?
No, just encourage labs. If they have any questions, they're more than welcome to reach out to me. I'm always happy to talk carbapenemase. As you can tell, I'm in my happy place right now. So yeah, I'm happy to take any questions from others.
That's great. Thank you very much, Trish. This has really been a great conversation. I thank you for this interesting and practical guide to lab detection of carbapenemases. Listeners can find both of the articles that we touched on at JCM's website, which is jcm.asm.org. And thanks for listening.